Hi, my name is Tal. I am a master's student in Josh Pusey's lab at William & Mary, and I'm studying a hybrid system that involves polyploid monkeyflower species that were actually introduced to the UK. So polyploidization and hybridization, they've widely been described as key evolutionary processes in the diversification of the angiosperms and their adaptation to climate and other environmental changes, as well as facilitation to invasion. So the genus Mimulus, or monkeyflowers, is often used as a model for studying local adaptation. Here we're using it more specifically to study adaptation with polyploidy and with hybridization. So this species has a lot of, these species have large life history traits and over a large environmental gradient. And so the system we're looking into involves these two species, Mimulus cutatus, which is a diploid from Western North America, shown in blue, and Mimulus luteus, which is a tetraploid from Chile, shown in green. And both of these species were actually introduced to the UK in the early 1800s, where they quickly naturalized within decades across the UK, and due to this novel concept, actually produced a hybrid. So this is Interploidy hybridization. Each of the parents are of different ploidy. So a haploid gamete is donated from the diploid cutatus, and a diploid gamete is donated from the tetraploid, producing this triploid offspring. And due to the uneven chromosome set, it is sterile. However, Mimulus reversi has been able to produce clonally through clonal propagation and is widely spread across the UK. So at some point along the way, on at least two independent occasions that are known, Mimulus reversi underwent a whole genome duplication, whereby it doubled its triploid genome to a even hexaploid genome that, now that it's even, is actually fertile and able to produce gametes. So what we see is this new hexaploid species, Mimulus peregrinus, which is considered reproductively isolated and fertile. So from this initial introduction of two species, we now actually have two additional species that are exotic in the UK. So this system is useful for, it's ideal for studying polyploidization, hybridization, because it has occurred quickly and very recently to where we still have the existing progenitors and some of their original populations. And we have genetic data on these parent species. Additionally, the genome duplication and the hybridization events actually were replicated multiple times in nature, and we can recreate them in the lab to study them a little more closely. So this allows us to address big questions on the mechanism of hybridization and polyploidization, as well as the genomic consequences of these processes, and the resulting phenotypic consequences, and ultimately what the fitness effects on the organisms are from these processes. So I'll be focusing mostly on the mechanism. And so when this hybrid was first studied, what was observed was this strong asymmetry with the germination of the seeds. So anytime we see, so the mom will always be on the left and the dad will always be on the right. So what we see is that when the diploid cutatus is the mom and the tetraploid luteus is the dad, looking at germination over a period of days, we see that while this hybrid cross has lower germination than the parents, it's actually much greater than the reciprocal hybrid cross, where the tetraploid luteus is the mom and the diploid is the dad. And this is actually unusual for reasons I'll get to in a minute. So we wanted to see if this held up in nature. And so using genetic sequences from maternally inherited chloroplasts, maternal haplotypes of cutatus and of luteus were created. And 114 individuals from 13 populations of Emberbirdsi, and 27 individuals from 3 populations of Peregrinus actually grouped with the maternal haplotype of Butatus, the diploid, whereas only 3 individuals from one population of Luteus grouped with Luteus. So we see the same asymmetry in nature. And so this kind of asymmetry and seed viability has been widely documented in interploidy hybrids, but typically in the opposite direction, where in the maternal excess direction, where the tetraploid is the mom. And this has been attributed to genomic imprinting, or the dominance of expression from one of the parents in the progeny. And this, in this case, occurs in the endosperm. So as a reminder, the seed has 3 genetically different parts to it. It has the embryo, which becomes the organism. And the seed coat is maternal tissue that holds in and protects everything. And the secondary tissue, the endosperm, which is rich in carbohydrates and proteins, and actually and it provides energy to the developing embryo. So what's unique in angiosperms is that this endosperm is actually fertilized in addition to the embryo. There's a second fertilization event. And it also has a higher ploidy level than the embryo, and that is typically due to the maternal genome. So we see this maternal excess, and so that has been explained as an adaptation to sexual conflict between mating strategies within the endosperm. So if we imagine a scenario with 3 different pea plants, we have one mom and one dad. In any given pea pod, the mom may have several seeds that are fertilized by different dads. So, but every single one of those seeds are going to be equally related to the mom, 50% related. So she actually wants to spread her resources evenly to all of her seeds to give them equal chance of viability. However, the dad, if we look at the seed on the bottom left, we see that it's 50% related to dad 1. But the seed next to it is actually 0% related to him. So he wants to selfishly pull for resources into the seed that he fertilized. So through this arms race, what has happened is that the mom actually has double the genomic content in the endosperm to effectively silence the selfish dad genes, or cause imprinting. However, if we have a scenario where the dad is a tetraploid, now he's bringing in twice as much genetic information. We have a 2-to-2 ratio within the endosperm instead of a 2-to-1 ratio. And we don't have that effective silencing. We don't have that mitigation of the sexual conflict. And so these seeds, typically the endosperm over-proliferates and they're inviable. However, on the other hand, if the mom is a tetraploid, now we have a 4-to-1 ratio, and while these seeds may still have some abnormal development, a little bit of under development of the endosperm, they're typically viable. And so we see this strong asymmetry in most described interploidy hybrids, where the tetraploid is the mom, and this is attributed to sexual conflict. However, once again, in our system, in Mimulus, we see this opposite asymmetry, where the diploid is the mom. So the question is, why is it different? What's driving this? What do we not know about sexual conflict and about asymmetry? Because this can dictate the genetics of the hybrid, particularly maternally inherited genomes. So when we looked at the seeds, what we noticed is that the parent seeds of luteus and of Butatus were round and plump. So these are about a ½ millimeter long. And the viable hybrid seeds, where the diploid was the mom, they were smaller and they were shriveled. The inviable seeds were actually flat and curled up on themselves. So the next step was to look at the inside of these seeds and what was going on with the different tissues. So I studied these crosses, the parental species, the diploid Butatus and the tetraploid luteus, And then each of the reciprocal hybrid crosses, the viable one, where the diploid mutatus is the mom, and the inviable one, where the tetraploid luteus is the mom. And I collected seeds over a period of 2 to 13 days after pollination, or DAP. So that was the developmental period. And I embedded these seeds in resin and then produced half-micron sections using an ultramicrotome to get these clear images where we can see the different parts of the seed, such as on the top, the outer seed coat and the inner seed coat. We have this endosperm here within the seed coat, and then this large embryo in the middle, and those are its 2 cotyledons on the end of a dicot. And then this little, the micropyle where the pollen tube actually enters. So what we see is across the 4 different crosses over a developmental sequence. Mutatus will follow our typical angiosperm development. We're at 5 days after pollination. We see what's called the globular stage of the embryo. At 8 days, it's past the heart stage where the cotyledons start to show. And by 11 days, it's mature and in this torpedo stage. So luteus, on the other hand, is a little slower than mutatus and is actually still at the globular stage at 8 days, while mutatus has passed that. Now looking at the viable hybrid, what we see is that it looks like the embryo actually follows the developmental pattern of its mom, mutatus. However, what's different is we see early on the endosperm isn't developing quite as well. It's constricted, it's much more confined within the seed, and that is in turn affecting the embryo. And the inviable hybrid is actually just completely, there we see no clear tissue development whatsoever within the inviable hybrid, and so this is not what we would expect from sexual conflict. But this closely matches the seed morphology with the round and plump seeds, and of the parents, the shriveled viable hybrid and the flat inviable hybrid. So to summarize these, we see that luteus has a slower developmental period than mutatus, and the viable hybrid follows the pattern similar to mutatus, its mom, but it is the embryo and the endosperm don't develop as well. And the inviable hybrid has complete failure at the earliest stages of the endosperm and the embryo. And so this matches our internal endosperm. So we've been able to describe what is happening within these seeds, and we see that something is not what we would expect from sexual conflict and from the typical patterns, but we haven't explained them yet. So if we take a little closer at the genomics, what we see is that luteus, instead of having 4 identical sets of the same genome, it has 2 unique subgenomes that independently segregate during meiosis. And so what we think is that the perceived ratios of the parents' genomes within the developing hybrid are altered because of that. And so we think that there is we're hypothesizing that there are subgenome-specific patterns of imprinting, and that this is what is driving asymmetry. So I've been able to successfully isolate embryo and endosperm, and so the next step is to sequence RNA to sequence the whole transcriptome from these tissues and identify imprinted genes specific to the subgenome and differences in the patterns of imprinting through these different processes. So I'd like to thank my advisor, collaborators.